So our next topic in this class is plasma membranes. In this video, we're going to go through the components of a plasma membrane and think a little bit about how they affect its function. So our plasma membrane is made up of 4 different types of molecules. We're going to see phospholipids, cholesterol, proteins, and some carbohydrates. These make up basically a mosaic, so like a tiled floor. These molecules are arranged next to each other in a fluid structure. As you saw in the previous video, the membrane is allowed to move up and down, a little shifting between these various pieces that make it up, which is going to be important as we start to talk about things that can move through the membrane. So the basic role of a plasma membrane is to control what comes in and to control what goes out, whether we're talking about a cell or one of our organelles. So you're always going to see these membranes at the borders of our cells. That's what you're drawing when you're drawing a little circular cell, or when you're drawing your little shapes that make up the organelles, which we'll get to later this week. And that membrane is controlling what enters and exits the cell. At the end of this week, we'll see that some of the components of the membrane also affect how that cell is able to respond to various signals in the external environment, and also affect how it's able to attach to nearby cells. So our first super important component of a plasma membrane are our phospholipids. So hopefully we remember from our macromolecules video that a phospholipid is a type of lipid, so that's our macromolecule group. And it has a polar part, which is made up of a phosphate group, potentially some other polar functional groups attached to that, a little glycerol backbone, and then 2 fatty acid chains, like these little legs, these feet here. So we can refer to these 2 parts of our phospholipid as the hydrophilic head. That's because this phosphate part of the molecule, because it is polar, is going to be attracted to water molecules. Remember that when something's polar, that really means that we're accumulating slight charges. So here you can actually see a little negative charge here on this phosphate group. So that's going to be attracted to the slight charges within a water molecule. Our other end of the phospholipid is our hydrophobic portion. So these 2 fatty acids, which can be either saturated or unsaturated, doesn't matter, so straight or bent, are going to repel water. They're afraid of water. And that's because they're non-polar. So they're just made up of a bunch of carbon and hydrogen linked together. There are no charges in that region of the phospholipid. So phospholipids naturally arrange themselves in a bilayer because of these properties. So whenever we drop a phospholipid in water, the phospholipid is going to try and arrange itself so that its polar head, that hydrophilic head, that phosphate part, is going to face towards the water, and it's going to try to protect those little hydrophobic tails. So the way we see this form in a membrane is that we get 2 layers of phospholipids. I'll put them this way because this is how they are in the drawing. So on the outside layer, we're going to see a bunch of phosphate heads here. And then their non-polar fatty acid tails are sticking down. So say this is our outside environment containing water. That water is touching our phosphate heads. They're polar. That's good. Those polar heads are hydrophilic. Then we have our hydrophobic tails facing inward. But in order to also protect those hydrophobic tails from the water inside the cell, we're going to have a second layer of phospholipids. So that second layer of phospholipids is basically facing the opposite direction. So we're going to have those hydrophobic tails touching the hydrophobic tails of our first layer. And those internal sort of phospholipids are going to have their polar heads facing the inside of the cell, so facing the cytoplasm. So we have 2 layers, and we basically have a double layer of fatty acid tails here in the center of our bilayer. So we call this the phospholipid bilayer. Bi means 2, layer, 2 layers. So here we have a sheet made up of our 2 layers. So here we can see how it looks in a sphere. So we see that we have polar heads on the inside and on the outside. And then we have this sort of internal part of our membrane, of our phospholipid bilayer, that's made up of this double layer of non-polar fatty acid tails. The second component we're going to see is cholesterol. And cholesterol is really going to be found in animal membranes, but since this course is geared eventually towards getting you through A&P, cholesterol is going to be important to understand for its role in cells. So this is a cholesterol molecule. And hopefully you can recognize what type of macromolecule this must be from its structure. We see it has these 4 rings pressed together, which means it's a steroid. So cholesterol is actually a second type of lipid that we're going to see in the plasma membrane. Now, it's found in only animal cells, and its main role is to affect the fluidity of the membrane, so to affect how much that membrane is able to move. Usually, cholesterol makes membranes firmer, so it makes them a little more rigid. But in certain cases, phospholipids can start to lock together, and in those cases, it's important to have cholesterol there, breaking them up, to stop the membrane from sort of crystallizing. So in certain rare cases, it also prevents membranes from becoming too firm or too rigid. Now, there are other factors that affect fluidity like temperature and the type of phospholipid, but we don't need to get into those right now. Our third component of plasma membranes are our proteins, and these are going to be really important. So there are 2 basic types of protein that we'll see in the plasma membrane. The first are our integral proteins, which we'll also sometimes call transmembrane proteins, because they go through the whole membrane. So they have a part on the outside of the cell, which we call the extracellular part. And they'll have a part on the inside of the cell, which we'll call the intracellular part. And there's also a portion that goes directly through, so sitting between those non-polar fatty acid tails of our phospholipids. There are also peripheral proteins, and they stay on one side of the membrane. We're really going to focus on the actions of integral proteins in this course. So let's zoom a little more on an integral protein. Now, the reason an integral protein can go all the way through the plasma membrane and all the way across the phospholipid bilayer is because we remember when we have proteins fold, we go from our amino acids in that primary structure forming a polypeptide with their peptide bonds, right? And then we have hydrogen bonds pulling that polypeptide into our alpha helices and our beta pleated sheets. And then we start to have a 3-dimensional structure because we have all these different R groups, all these different side chains with different chemical properties. They pull our protein into a 3-dimensional shape. And because there are so many different amino acids with so many different chemical processes, some parts of that protein are going to be hydrophobic, other parts are going to be hydrophilic. This is true of our integral proteins, and what happens is that we have a hydrophilic region on the outside and on the inside of the cell. And then we have a hydrophobic region, which allows the integral protein to sit in that same space as our nonpolar fatty acid tails. So this can happen because this doesn't stop this whole central layer from being nonpolar, from being hydrophobic. It can stay there because it also doesn't like water, this part of the protein. Our final component of a plasma membrane are our carbohydrates. So here it says oligosaccharide carbohydrates. All that means is that they're carbohydrates that kind of like stick on to other parts of our membrane. So we're going to see basically 2 types, And they're just named for whether the carbohydrate is stuck to a protein or stuck to a lipid. So if it's stuck to a protein, here in blue are our proteins. Here's an integral protein. Here's a peripheral protein. And here we can see that one of those other proteins in the membrane has these structures here. And if we think about it, we should be able to recognize that that's a carbohydrate because we have a long chain of rings and we can see some branching. So we wouldn't see that in any other type of macromolecule. The only other place we might see sort of bead-looking structures really would be our proteins, but then we wouldn't have this branching. Okay, so this is a glycoprotein. These are sugar rings stuck to the outside of a protein. This is a glycolipid attached to one of the phospholipids itself. So it's more little sugars attached to the outside of a lipid. And the function of these carbohydrates is really to allow cells to recognize each other and partially to allow them to attach to each other.